Thank you all for staying to the last. What I'm going to be doing is talking about some bacterial work, and that's been highly underrepresented in this meeting, actually. So I'll make up for that. So we work with the Initiator tRNA and its roles. And are we moving here? Okay. Yeah. So before I go and talk to you about Initiator tRNA, let me introduce to you the Initiator tRNA itself. So we work with E. coli, and in E. coli, there are actually 4 genes that code for Initiator tRNA. 3 of them are at this one locus, MATZWV, and the 4th one, a little away from it, is in this locus. All of these tRNAs make roughly the same kind of tRNA. There's a single mutation in this gene right there. We will not worry about that right now. But what you see here in this clover leaf structure of this Initiator tRNA, there are 2 regions that I have highlighted. One is in the anticodon stem, and the other one in the acceptor stem. Anticodon stem has these 3 GC base pairs, which are highly conserved in all domains of life. And if you look at the acceptor stem, there is a mismatch at 172 position. And this is really very important for many things. And there are sequences at 2nd, 3rd, 4th, these positions, which are all important for formulation of this tRNA. The mismatch helps in the formulation of this tRNA. It also helps in avoiding this tRNA from binding to EFTU, as well as yet another function, which is an enzyme called peptidyl tRNA hydrolase. So formulated tRNA looks like peptidyl tRNA, and peptidyl tRNAs have to be recycled, and that recycling happens with the enzyme called peptidyl tRNA hydrolase. So this mismatch actually also avoids unnecessary hydrolysis of this formylmethionine from this tRNA. So a single mismatch can actually do many things, and so it's really very important. Now, the only thing that I should also tell you is that the formulation is required only in bacteria as well as eukaryotic organelles. So these 2 regions are really very crucial for Initiator tRNA. And if we now look at the participation of tRNAs in protein synthesis, what you will notice is that the elongated species of the tRNA is amino-acylated by one of the 20 amino acids. The amino-acyl tRNA then binds to elongation factor TU, and then it is taken to the ribosomal A site. Whereas the Initiator tRNA is amino-acylated with methionine tRNA synthetase. The amino-acylated tRNA is then formulated by the enzyme called formylase, and FMAT tRNA then goes to the ribosomal P site. Now, there are many versions, where some people think that I have to carry this tRNA to the P site, but I guess the more likely scenario is that all of these 3 factors are actually bound to the small subunit to begin with, and so this actually captures the FMAT tRNA onto the ribosome. So now, as I was mentioning that, this tRNA goes to the P site, and this is actually the only tRNA that under normal conditions will go to the P site. The elongator will go to the A site. What people have conventionally thought is that this tRNA does not bind to elongation factor TU, and therefore it does not participate in elongation. But what I would show you is that, if this tRNA accumulates here and it is not taken to the formulated species fast enough, then some of this tRNA can actually bind to EFTU and can actually participate at this step of elongation. And as I mentioned to you earlier, that this mismatch is really quite important for avoiding this step, but when you have accumulated quite a bit of this methionine charged species, which is not formulated, can actually go to the A site as well. So this can indeed go to that step as well. Likewise, there is a thought that the elongator tRNA does not participate at this step of initiation. And this also, I would like to convince you that happens when you have low amount of formylmethionine tRNA. Under those conditions, the amino acyl tRNA can actually participate at this type of initiation as well. So these are the sort of canonical functions, but besides their canonical function, initiator tRNA and IF2 in initiation, I would also like to show you that they do participate at this type of ribosome biogenesis as well. So this is the picture that I've taken from Goalaji's work. And what you will see here is, this is a small circle is 30th subunit and big circle is 50th subunit. So the initiation factors are actually bound to the 30th subunit. And it can actually go two ways. One, of course, this 30th subunit can bind to the messenger RNA, and then it can bind to the initiator tRNA to give you this. And this will, in most of the cases, will happen when Shine-Dalgarno sequence is strong. And in the cases where either it is weak or actually is not there in the messenger RNA, the cell may actually follow this path where the 30S is first binding to the initiator tRNA, and with the help of the initiator tRNA, it can help recruitment of messenger RNA, and at the end actually come to a situation where the initiation factors, initiator tRNA and the messenger RNA are together. So this is a sort of 30S pre-initiation complex, and this 30S pre-initiation complex then goes through a conformational change, and which is actually the most time rate limiting step here. This forms a 30S initiation complex. It then joins with 50S subunit, initiation factors are removed, IF2 GTP is hydrolyzed, and IF2 is also released. And now this complex, 70S complex, is ready to go into the mode of elongation. So this is all done through in vitro work. And we've been interested in looking at the initiation aspects in vivo. And so for that, we have developed a very simple system. And the system is that we take a reporter gene of chloramphenicol style transferase, change the start codon from AUG to UAG, and also make a corresponding change in the anticodon of the initiator tRNA from CAU to CUA. And this system actually works quite well. The reporter gene is translated, and the cells actually become chloramphenicol resistant. The only difference that you will find, because we made the change in the anticodon, this tRNA is now amino acylated with glutamine, and the glutamine is actually formulated just as efficiently or nearly as efficiently as methionine is. So we don't really face any problem there. Now, one of the things that we were quite interested in trying to look at the importance of the 3GC base pairs. So here what we do is we change the 3GC base pairs to a sequence that is found in the elongator species of methionine tRNA. And we call this as a 3GC mutant tRNA, and many times we call it as a 3GC tRNA. Even though the 3GC are not there, we call it 3GC tRNA. When we take this tRNA, it actually does not initiate from this UAG codon, and the cells become chloramphenicol sensitive. And if you look at the amino acylation formulation, there is absolutely no difference compared to this tRNA. So this is amino acylated and formulated just as efficiently as this one is, yet it is not able to participate at this type of initiation. And that should not be any surprise, because we are actually changing one of the very, very conserved feature of the initiator tRNA to that is found in the elongator tRNA. So now what do we do with this dead tRNA? In fact, this is something that we wanted to develop. And the reason we wanted to develop this was so that we can do set up a genetic screen and ask a question: are there things in the cell that would actually rescue the defect of this 3GC mutation in the tRNA? And so the system is very simple. Again, we have the cat amber where the chloramphenicol start codon has been changed to UAG. This will normally not initiate, and the cells will be chloramphenicol sensitive. So now if we have made a mutation in the cell such that it actually rescues the defect of this 3GC base pair, we would now start initiation from this UAG codon and will convert the chloramphenicol sensitive cell into chloramphenicol resistant cell. And by doing so, a postdoc fellow in the lab, Dinesh, who is actually a professor in Oklahoma right now, he actually isolated many of these strains, which we call them as suppressor strain. And we have pretty much characterized all of them. And looking at many of these features, what we now understand is that the formulation of the initiator tRNA is important mostly for the initial binding of the tRNA to the 30S. Subsequent to this step, it probably has absolutely no role. And one of the reasons we can say so is that if you take a bacteria where formulation has been avoided by, for example, deleting FMT gene, which codes for formulase, and now you start overproducing this tRNA, it actually compensates for that defect altogether. And so that kind of a rescue can happen only if the role of formulase or formulation was right at the first step. If it was a subsequent step, the effect will not be so much. And in fact, if we try to overproduce a tRNA which is defective in the 3GC base pair, it will not do that. So the role of 3GC base pair actually is more in these conformational changes that I was talking to you based on that in vitro analysis, that there is a rate limiting step of conformational changes that gives rise from 30S pre-initiation complex to 30S initiation complex. So 3GC base pairs are involved in taking this tRNA or transiting it from the initial binding to the final binding in the PP site, and the role of formulation is right at this step. So this sort of, we understood from very simple experiments actually. We could also combine it, we did combine it with northern blot analysis also to understand where the tRNAs are. So now if we look at this pathway once again, so you have the tRNA, which is amino acylated, and it's then formulated, and it goes on for initiation. What you will notice here is that these two things, methionine and N10-formyl tetrahydrofolate, which are required for amino acylation and then formulation, they're actually coming out of this one carbon metabolism. So 10-formyl tetrahydrofolate you have here and methionine over there. And so one of the things that we noticed was at a good number of the suppressor strains that we got were actually either defective in folate dehydrogenase, which would actually limit the amount of 10-formyl tetrahydrofolate, or actually one of the strains that we found was actually having mutation in this FMT. And so the FMT activity was actually dead. And yet actually I'll tell you that, when we look at this guy, this AMBA codon has been placed in such a way that it trims the protein by 42 amino acids at the C-terminal. And we know that even if you delete 20 amino acids from the C-terminal, the enzyme becomes dead. And so this actually, the enzyme that it will normally produce would actually be dead. But in this strain, we find that the tRNA actually gets formulated. And I'll come to that one also. So what you notice here is that limiting the amount of this will then allow this tRNA to actually even participate at the step of elongation. And we notice that it binds to EFTU, it can suppress the termination codons in the reading frame. So now we have a situation where a single tRNA can participate at the step of elongation, can participate at initiation. Now, if you think about it, in mitochondria, we have precisely the same situation, that the single tRNA initiates as well as elongates. So the question that we then asked was, can we create a situation in E. coli where a single tRNA will sustain the functions of both initiation and elongation? And can we do it without tampering with FMT or N10-formyl tetrahydrofolate? And if you look at this scheme here, this we don't want to tamper, this we don't want to tamper. The only thing that we can tamper would be this tRNA itself. And as I mentioned to you, that the acceptor stem region is very crucial for formulation. And we have already characterized many of these mutations will compromise the tRNA for formulation. And so we could actually use these mutant tRNAs and ask a question that if we use these mutant tRNAs, can we now delete these three genes, this single gene, and both of the elongator tRNA genes? And Ashwin in the lab did this work, and he was indeed able to do that. He was able to get rid of all of these 6 genes. And what he found was that this particular mutant of the initiator tRNA, which has U1, U3, A70 mutation, was actually the best to sustain the growth of E. coli. And if you think about it, if you look at this here, this is very similar to what is found in mitochondria as initiator tRNA. The only difference here is that rather than AU, we have UA here, but both weak base pair here, and this third base pair is UA anyways. So this nature has come up with a situation in mitochondria where a single tRNA can do both the jobs, and we can actually reproduce that in E. coli as well. So now coming to the point that I mentioned to you, that in FMT, MR274, which is clipping it off by 42 amino acids, how do we get formulation of the tRNA going on? And this turns out to be an autoregulatory system here. In the cell, only thing that we have really is this tRNA, which has the anticodon, which can actually read the UAG codon. Normally, this tRNA, whether it is amino acylated methionine or glutamine, will get formulated and will not participate at this step of elongation. But now, if to begin with, this FMT is not active at all, let's say, we create a situation, think of a situation where this is not being produced, this tRNA will accumulate. Now, this tRNA can bind to EFTU and then suppress this termination codon and give you FMT. Now, if you suppress this, you produce FMT. When you produce FMT, you start formulating it, and now it goes into the initiation pathway. So even in this system where we have FMT having a suppressor or having an EMBER mutation, this can create a situation where a tRNA can now participate at both the initiation and elongation steps. So now the other thing was that, are these things of any use? If you look at this one carbon metabolism, there are many drugs that are used to kill bacteria, whether they are sulfur drugs or trimethoprim, and they will work at these steps. And so what Kuldeep did was, we had actually 3 alleles where folate dehydrogenase was having mutations. And if you now put trimethoprim, so this is the situation with the wild type strain. This is happily growing at this trimethoprim situation. But these strains which are deficient in folate dehydrogenase activity don't grow, even at 0.5. And even at 1, of course, they don't grow, but wild type grows. And a control situation, they roughly grow the same at 0 trimethoprim. So it's quite possible that to design, as of now, we don't know of inhibitors of folate dehydrogenase, but if we design inhibitors of that, it essentially becomes a double kill, and the one carbon metabolism can be effectively downregulated to come up with the antibiotic resistance problem. In fact, these guys have quite a bit of resistance developed in the bacteria. So I think this would be one solution to that problem. Now, there was also this question that was raised when Deepa was actually talking about the work. So last year, actually, was a student in my lab, and then she went and worked with Deepa. And what she did actually was that if you change the start codon of uracil DNA glycosylase gene, she wanted some enzymatic assay along with other cat assays. So she developed a strain where the 3 tRNA genes were deleted, and this uracil DNA glycosylase gene was deleted. And she took the uracil DNA glycosylase gene, changed the start codon to any of these codons, and she had the tRNAs, which natural tRNAs, elongated species, which will pair at that start codon. And she wanted to then ask a question, what happens? So in the strain which is deleted for 3 tRNA genes, you see that the protein initiates so much. Now, if you overproduce initiator tRNA, this initiation actually comes down. From there to there, it comes down, or from there to there, it comes down. And this situation you see with many of these elongated tRNA. Now, this is in the log phase culture. And interestingly, when she takes the stationary phase culture, you see the initiation levels with elongated tRNAs actually increase quite a bit, but they all come down. The moment you overproduce initiator tRNA, they all come down. So there is this connection that if you've got less amount of initiator tRNA, elongators can also actually go on and participate at this step of initiation. So now let me come back to the other aspect that I was referring to, the role of initiator tRNA in ribosome biogenesis. So a very earlier student, Suman, who was working on these suppressors, and all of these suppressors actually had a mutation in the locus of initiator tRNA, which has 3 genes, which is called as FMAT1 locus. And the mutation was such that the transcriptional level of this was really minimized to almost non-detectable level. So when she actually plated this bacteria with these suppressors, and then she of course created a defined mutation by deleting this locus, what she noticed was that this strain, which was deleted for 3 tRNA genes, was cold sensitive. Now, cold sensitivity is generally associated with ribosome biogenesis defect. So obviously the question was that, is initiator tRNA involved in ribosome biogenesis? And just to introduce to you, so in bacteria, you have a single transcript that is having all of these genes, and then they are cut at these positions, and then ribonucleases come and trim off the extra sequences at the 5' and the 3' end. And of course, this is Nomura scheme there. What you will notice here is that when all of this has assembled, there are still some sequences at the 5' and the 3' of 16S rRNA, and likewise at 23S rRNA, which need to be trimmed. And that trimming, final trimming, happens with many of these ribonucleases that are indicated here. So what Sunil, who was at the meeting until last week, what Sunil did was, he did few experiments. And what he wanted to see was really what is happening. Is it really ribosome biogenesis problem? So what you will notice here is that based on simple experiments of northern blot analysis, you could design a probe which will detect the extensions or detect the mature form of the ribosomal RNA. And what you notice is that in the deleted strain, the level of the 17S rRNA, which will be because of these extensions being there, he finds more of the 17S. That means in the absence of initiator tRNA or limitation of an initiator tRNA, this final trimming does not happen. So then he also did another genetic experiment here, and what he showed that this trimming, final trimming, there is genetic interaction of the initiator tRNA with many of these ribonucleases which are involved in here. So what we mean by genetic interaction is that if you delete these ribonucleases, the problem is enhanced, and if you overproduce these ribonucleases, the problem is solved. They are rescued. So that was another experiment. And yet another experiment that he did, he actually used a situation where he was using the wild type tRNA and of course, start codon, which is in this case AUG. This will be a normal growth. Now, if in this particular bacteria, rather than taking wild type tRNA, he produces this 3GC mutant such that it will not initiate now because this 3GC doesn't really, mutation doesn't allow. It actually compounds the problem. There is inhibition in the growth. And if he now takes the same tRNA but changes the anticodon to CUA such that it will not pair with this AUG, the problem is relieved. So again, he showed that the process of initiation itself is involved in this ribosome biogenesis issue. So what he was able to show was that we have this 30S, where this RBFA is the factor which is working towards the end. And this RBFA is actually removed from ribosome by another biogenesis factor called RSGA. This is the GTPase. So you get a situation like this, and you see these are extensions at both the 3' and the 5' ends. The initiation factors are binding there, initiator tRNA, mRNA come there, 50S unit comes. And it is actually at this stage, these extra sequences are trimmed away. And so in the pioneering round, really, somehow this initiator tRNA, when probably the 70S complex is formed, somehow triggers that the ribonucleases now can trim away these extra sequences, and you get the mature ribosomes. Now, this story got a little bit more involved in the sense that another student, Kuldeep, he was looking at yet another suppressor strain called A18. And in that strain, the mutation was in RLUD, which is a pseudouridine synthase. And what you see in the pseudouridine synthase is you cleave this bond and make this bond here. So it's over there. And if you look at this RLUD protein, the mutation is right at this 3' end where the E265 residue is changed to lysine 265. And this RLUD actually works on the helix 69, which is a very crucial helix. It actually docks onto the helix 44 of 30S and makes that bridge B to A, which is very, very important bridge in ribosome. So all of these three positions are actually modified by the single enzyme RLUD from U to pseudo U. What Kuldeep noticed was that steady state levels and in vivo in vitro catalytic activities of this mutant were unchanged. So this enzyme was expressed in the cell exactly at the same level as the wild type enzyme was. And if you look at the catalytic activity of these two proteins, mutant and wild type, they are also identical. Overexpression of catalytically dead RLUD also suppressed the phenotype of this 265K. So the phenotype of this was that it allows initiation with 3GC mutant, and if you overproduce the catalytically dead RLUD, that also rescued that defect. And the role of E265 is independent of the pseudo-U synthase activity, which is apparent from that point as well. And this actually then led us to think that there must be a second function of RLUD protein, much like what Rajyaguru was talking about his protein, that there is a function of this protein which is independent of its catalytic activity. And he did many other experiments, and what he actually eventually added to this pathway was that what we assumed earlier, that this RBFA is released completely by RSGA, is not really the situation. There are residual levels of RBFA on 30S, and it goes all the way up to here. And at this point, RLUD actually removes whatever that residual level of RBFA is. So initially we removed entire RBFA at that point, but actually entire RBFA gets released at this point. So that was the contribution that Kuldeep made in this ribosome biogenesis or maturation scheme. And then he actually looked at the literature. That always helps actually. And there was a study done by Eric Brown in 2008. And what he had done was he had looked for the suppressors of what was called as YJEQ. And this protein is nothing but RSGA. So he had isolated suppressors which would rescue the defect of RSGA or compound the defect. And what you find is that one of the genetic interactors here is RLUD. So this also gets connected quite well with the study that Eric Brown had done. So moving on to what Eric Brown has been doing, what Eric Brown found was that he was screening for compounds, known drugs or compounds, which will compromise ribosome biogenesis in E. coli. And one of the molecules that he found in that screen was lamotrizine. And the target of lamotrizine, IF2. And so because everything is fitting in there, we thought, okay, let's look at it. The mechanism, of course, was not clear, but I think we know the mechanism now. So this is the IF2, and there are several domains here, but domain 2 is of importance to us. And what he noticed was, this he reported, that if you actually delete a certain stretch of amino acid from domain 2, this IF2 no longer binds to lamotrizine, and lamotrizine is no longer toxic to E. coli. So this is shown here. So if you take E. coli strain is growing, overexpression of IF2 actually is somewhat toxic. So you see growth defect here. And when you take lamotrizine, IF2 is toxic, but when you take the strain, if you take the construct which is deleted for 17 amino acid right there, this is now no longer toxic with this lamotrizine. So this makes sense. Wild type IF2 confers the effect of lamotrigine toxicity, but the mutant strain, which is defective in lamotrigine binding, does nothing here. And we can also see that, in wild type strain, as you increase the lamotrigine concentration, you see that there's more accumulation of 17S rRNA. But if you take IF2, which is deleted for 17 amino acid, you don't see that increase. So that also fits quite well. So we wanted to understand, look, if it is, lamotrigine, what exactly it is doing. And so our immediate thought was that it must be affecting either the GTPase activity or the initiation activity directly. But as you can see that when you compare the GTPase activity by taking lamotrigine at different levels, whether you do the wild type IF2 or delete it for 17 amino acid, nothing happens. You can also look at the initiation activity, whether you initiated with the 70S initiation complex or 30S pre-initiation complex, neither of them is affected. So it doesn't affect initiation, doesn't affect GTPase activity. So what does it actually do? So what Sudhir actually did, this is the work carried out by Sudhir only, he did the polysome or the ribosome, polysome profiles here. And you will notice that there is disome, trisome in wild type control. As you take lamotrigine into it, the level of this decreases. But if you take the resistant strain, that level remains there. And whether you take lamotrigine, nothing happens. Now, if you look at the RBFA levels in these profiles, wild type has some amount of RBFA. But when you add lamotrigine, this RBFA level increases. And remains roughly the same in the strain that is resistant to lamotrigine. So then the thought here is that lamotrigine targets IF2, but interferes with RBFA release. So how? And then we moved on. Yet again, we went to the screen of Eric Brown. And what you will notice here is in one of the multi-copy suppressor screens, what he finds is that he can suppress the RSGA phenotype by overproduction of IF2. And so the idea now for us was that are IF2, RSGA, RBFA, lamotrigine connected in some way? And so lamotrigine, does it interfere with the function of RSGA? And so this is then experiment done with the strain which is deleted for RSGA. And what you will notice here is, well, you can just look at this one. So deleted for RSGA, this is the empty vector. And as Raj was saying, that we should verify the published results, and that verified the published report that IF2 overproduction rescues it. Now, when we produce initiator tRNA, not much happens. But if we overproduce initiator tRNA and IF2 together, then the rescue is much better. So the effect of IF2, one of the effect of IF2 definitely is by participation at this step of initiation. But does it have anything beyond that also? And in fact, whatever we are noticing here actually agrees with the level of 17S, which is also quantified here, more, less, more, less. Yeah? So that fits quite well. We can also do the growth curves. And if you look at the wild type strain, lamotrigine compromises the growth. Delta RSGA, this is minus LTG, with LTG improves the growth. You take the initiation factor 2, which is deleted here for one amino acid, that is also resistant in fact, and you see nothing happens. So if you look at these profiles again, and the profiles here are, this is the delta RSGA profile. Actually, one of the phenotypes here is that 70S peak doesn't really come out that clean. And when you add lamotrigine, now you start seeing 70S peak. And this is somewhat similar to what you start seeing with IF2, which is the characterized suppressor of delta RSGA. So this fits quite well. And then what Sudhir did was, he looked at many of the suppressors that he had isolated, or many of the resistant alleles of IF2 that he had isolated. So delta 1 is one of them, delta 8 is another one, delta 17 is something that Eric Brown also saw, and there is a duplication of the sequence that also becomes resistant to lemotrizine. So all of these are actually lemotrizine resistant. And what Sudhir saw here was as follows, that IF2 overexpression rescues RSGA. And what he wanted to know, do these alleles also rescue? And what you will notice here is that the IF2 alleles which are resistant to lemotrizine are also rescuing the defect. Yeah? And so this really also seen by quantification here. And he also looked at yet another phenotype. And another phenotype is that when you have RSGA deficiency, this helix 45 is trimmed away. And you see that as a small band here. I'm not sure if many of you will be able to see, but it's right there. And this band disappears when you overproduce any of these IF2. So this also shows that IF2 is able to rescue irrespective of the lemotrizine binding region or not when you overproduce this species. So the observation that we have so far are as follows, that you have in delta RSGA, lemotrizine will rescue, and this rescue is dependent on the presence of lemotrizine binding region in IF2. And this is when you have IF2 on a single copy. But when you have IF2 in multi-copy, then it will rescue the delta RSGA effect independent of lemotrizine and lemotrizine binding region. So at low abundance of IF2, we require lemotrizine to rescue delta RSGA. At high abundance of IF2, irrespective of lemotrizine or the binding region, we are able to rescue delta RSGA. So does a conformer of IF2 substitute for the function of RSGA? That became the question, because that's sort of pointing us in that direction. And around this time, there was this study that came out in bioRxiv. Unfortunately, we still don't see this as published. But what they found out was that IF2, like many of the proteins that have been talked about here, has IDR, intrinsically disordered region. And they were also able to show that it will go into these bodies, liquid-liquid phase separation, LLPS that they call. And they can show it in lower temperature in bacteria also. If they tag IF2 with something, they can actually start detecting these aggregations. So then IF2 IDR promotes phase separation with RNA, cold temperature promotes IF2 condensation with RNA, and IF2 IDR promotes fitness during cold trough. So we then decided, maybe we thought, a hypothesis then is that, IF2 probably exists in multiple conformers, or at least two conformers, and maybe one of the conformers is helping that. And so the situation is as follows: when you overproduce IF2, naturally there is some conformation which might be doing the job of RSGA, but when you don't have enough of IF2, then you force it by putting LTG, lemotrizine, which will bind to IF2 domain 2 and then bring out some change in the conformation. So if you now look at the delta RSGA strain and overproduce IF2, which is what the authentic suppressor it was found as, perhaps an altered conformer of IF2 does the job of removal of this RSGA from the 30S ribosome and maturation of the ribosome. When you have wild type E. coli without overproduction of IF2, lemotrizine will do the same thing on this and will do this job. Now, in case of E. coli wild type, lemotrizine becomes toxic. And that can be explained by considering that RSG is already there. And RSG is doing a pretty good job of removing this RBFA from 30S and giving you, 30S, biogenesis going on. Now, if you actually bring in IF2 also to do this job and doesn't do as good as RSGA, then it becomes toxic. And this is basically what, now you bring in this IF2 and then try to bring it here. It is interfering with the role of RSGA and probably becomes toxic. Now, if this whole thing is correct, then what might happen is that if we actually isolate suppressors in RBFA protein such that they don't require any help, either from RSGA or from IF2, it should actually become independent. And this is basically what we find, that you have a situation where the delta RSGA, lamotrigine is rescuing it. Now, if you get this RBFA mutation like this one, and here, now it sort of grows even better. So perhaps the function of lamotrigine is to alter the conformation of IF2, which can now bind to the place where RSGA will bind and actually remove RBFA from the ribosome. And it is possible because they occupy the same site on helix 44 of 30S subunit. So this then finally what we think is happening here is that original story of Sunil, then Kuldeep coming up with the idea of RBFA being removed here by RLUD, and then Sudhir coming up with the picture that if you don't have RSGA, actually IF2 would also do this job. So this is the major conclusions here, that initiator tRNA formation drives the final step for ribosome maturation. IF2 plays a dual role. One is canonical role in initiation, and a conformer of it serves as a poor mimic of RSGA. So it's helpful to delta RSGA strain, but toxic to wild type strain. And the observation suggests that the major impact of lamotrigine is at the step of RSGA function. With that, let me finish. And the work that I presented to you has been done mostly by Sudhir, Jitendra, Kuldeep, and we had a collaboration with Suparna Sanyal for the initiation, in vitro initiation assays, which was done by Chandrasekhar Amandava. And the work in my lab is supported by these funding agencies. And thank you very much for your patience listening a bacteria story.
So I don't think there were hints of this in eukaryotes, of the idea that there's you essentially do final maturation steps as a check, but the idea that initiation factors do it seems like quite a new idea. And it like is your thought that it guarantees that everything is in place and everything works, and that's how you make the final decision? Is that the?
I think it will be necessary only at the pioneering step, as you can imagine. Right. So I think initial, I mean, I don't know, but I think that if the ribosome is not fully mature, when it is just being produced, it probably makes sure that it becomes a perfect ribosome before it goes on to make more proteins.
It's a really cool idea.
Yeah.
So, Umesh, RSGA is an assembly factor for the 30S subunit, right?
It is a biogenesis factor, yeah.
Right. So I have to remove this factor during the biogenesis. But you mentioned the RLUD should urease also do the same job, right? But this is a modification enzyme bind to the 50S subunit.
RSG is more efficient, yeah.
RLUD.
RLUD.
Yeah.
Okay, I think.
Urease, right?
Yeah. So what is happening, I think. Now, RLUD binds to helix 69. And I have a feeling that it remains on helix 69, there only. Mm-hmm. And so when the 50S subunit is trying to join, at that point, RLUD might be removing whatever RBFA is there and itself also going away. I don't know if I have a slide on that. Yeah, here. So what we are thinking is that this is your helix 69 there, and RLUD is bound there. And so when this helix has to dock onto the place, this is helix 44 is covered by RBFA. So when this is trying to join here, both the proteins probably leave. Only a hypothesis. I have not shown it, or we have not shown it. But this RLUD remains there for some time.
But the RLUD binds to the helix 69, right?
Correct.
In the 23S ribosome RNA, right?
Sorry, huh? 23, so that's right, yeah.
Right.
Yeah.
But in this model, I'm very confusing. So there is no 50S, I mean, right?
Oh, there is 50S. The 50S will come. So the final removal of RLUD is happening only when 50S unit is docking onto that
I
30S
see.
complex,
after
which is
making-
already bound with the initiation factors and tRNA and
I see.
mRNA.
I
Yeah.
see. Here, 69 close to the smooth.
Correct, correct.
I
Yeah,
see. I
yeah.
understand. Thank you very much.
Yeah. I mean, it's probably doing a lot of things. In fact, this is also responsible for removing IF3. Because IF3 is also bound there. So, this joining probably has a lot of purpose.
So do you think the mutant, I don't know, remember the number, does it not come off or does it like
The
this-
3GC mutant.
the one that, yeah, the RLUD mutant, do you think that is a more stable binder or? less stable?
RBFA mutant or RLUD? RLUD.
I think
So that is happening in the C-terminal domain, which is right here. So something it must be doing that it actually doesn't do it so well.
Yeah, I was wondering if it was like too stable and it couldn't come off. It
Yes,
fails to
yes,
come
yes,
off.
yes.
I just have a naive question, but can you, now that you know that this trimming is the first round of translation, the 17S to 16S.
Right.
In E. coli, can you estimate how many ribosomes have not been used yet or still in the 17S mode? And does that change in different conditions?
We have done the northern analysis, but those are two different probes. So we haven't bothered to quantify them. But if we can quantify those by using the same specific activity probe, we should be able to do that. Yeah.
Okay, so does the Lamotrigine bound IF2 shows better binding to 30S?
We haven't done those binding assays. And I think even Eric Brown has not done those. Yeah.
And another question is, so you showed in your first part of your talk that many elongated tRNAs can take part in initiation. Now, is that, in those steps, does it go through 30S or it is initiation directly at the 70S?
I think it goes to 30S only because, there are IF2 proteins which don't necessarily require formulation of the tRNA. So it's quite well known for Pseudomonas IF2. Okay. It doesn't require formulation. So because binding of the IF2 tRNA with the IF2 will be quite much required there. So I think it is happening at the level of 30S only.
Any further questions? Okay, let us thank Professor Umesh Varshney for this wonderful.
Thank you.